Soil bacteria build some of our most useful antibiotics on a molecular assembly line. It's a single enzyme complex, one of the largest proteins a cell makes, and it snaps a drug together one chemical piece at a time. A team at the University of Texas at Austin took that assembly line apart, rewrote it as swappable parts, and got it running inside ordinary E. coli. Here is the machine in plain terms. A polyketide synthase is a chain of enzyme modules, and each module adds exactly one small carbon building block to a growing molecule. Every module has the same core tools. One part grabs the next building block, one part carries the chain, and one part welds the new piece on. Some modules also carry trimming enzymes that reshape the piece before passing it along. At the very end, a release enzyme closes the chain into a ring. Run the whole line once, and out comes the core skeleton of a macrolide antibiotic. The specific assembly line here is the Pikromycin synthase from a soil microbe called Streptomyces venezuelae. In nature, it makes the antibiotic Pikromycin and a cousin called methymycin. The problem is that these bacteria are slow and awkward to engineer, and the assembly line itself is enormous, far too big to just paste into a friendlier organism and expect it to fold and work. There was also a deeper design mistake baked into decades of earlier attempts. When researchers first sequenced one of these assembly lines back in 1990, they drew the boundary between modules in what turned out to be the wrong place. For years, people cut and pasted modules at that line, and the swaps mostly failed. Around 2017, work on these enzymes showed the real seam sits a little further along the chain, so each module keeps the natural partners it evolved with. Cut at the correct seam, and a module behaves like a self-contained unit you can actually move. That correct seam idea is what this team built a factory around. The catch is that E. coli can only hold a stretch of engineered DNA up to a certain size before it becomes unstable, and the full Pikromycin assembly line is bigger than that limit. So the researchers, Takeshi Miyazawa, Ramesh Bista, and Adrian Keating-Clay, split the blueprint across two plasmids, two separate loops of DNA. One loop carries the front of the line, the other carries the back, and they engineered matching docking grips so the two halves find each other and snap together inside the cell. The two-plasmid trick does something clever beyond just fitting. Each plasmid has an insertion slot, so you can drop new modules into the middle of the line one at a time, like adding cars to a train. That turns a giant, fragile construction job into something modular and repeatable. When they assembled the refactored Pikromycin line this way, E. coli produced the core macrolide ring, a molecule called narbonolide, at about 49 milligrams per liter of culture. For a rebuilt mega-enzyme running in a foreign host, that is a real usable amount. But narbonolide is only the skeleton. It's a bare ring, and a bare ring is not yet a working antibiotic. To become one, it needs a sugar. Real macrolide antibiotics carry a specific sugar called desosamine hooked onto the ring. And that sugar is a big part of how the drug grabs onto a bacterial ribosome and shuts down protein production. So the team added a second set of genes to their E. coli, the enzymes that build desosamine and clip it onto the ring, plus an enzyme that adds a single oxygen atom at the right spot. With those tailoring steps in place, the same E. coli platform stopped making just a skeleton and started making actual named antibiotics: narbomycin, Pikromycin, YC17, methymycin, and 5 more related compounds. One assembly line, one added toolkit, and a small family of real macrolides, coming out of a bacterium that never makes them in the wild. At first, the antibiotic yields were low, and the reason was a traffic jam over sugar. E. coli uses the same kind of sugar-building precursor for its own surface molecules, so the cell was competing with the desosamine pathway for raw material. The team used CRISPR to knock out several of those competing routes, freeing up the precursor. They also restored the assembly line's natural docking grips and added an editing enzyme that clears stalled, mismade chains off the line. Together, those changes pushed the output of narbomycin up 58-fold to about 37 milligrams per liter. Now, the real payoff of building it modular. Because each station can be lifted out and replaced, the team could swap in modules borrowed from a completely different assembly line, the one that makes the drug rapamycin, and change the shape of the product on purpose. Swapping the second and third modules this way produced 3 brand new narbomycin analogues that had never existed before. When they tested them, one of the new compounds killed bacteria about as well as narbomycin itself. The other 2 were weaker. That is the whole point of a programmable line. Change a part, get a new candidate molecule, and read out what the change did. It's worth being clear about what this is and isn't. This is not a push-button machine that prints any antibiotic on command. When the team tried single module swaps across the Pikromycin line, most of them failed outright. Only one gave the product they were aiming for, and many working hybrids came out at lower yields. The oxygen adding step is still a bottleneck for some of the products. Reprogramming these enzymes is still hard, and there is no general recipe yet for making any swap work. What the team has really delivered is a workbench. They showed you can take one of nature's giant antibiotic assembly lines, cut it at the right seams, rebuild it inside a fast, well-understood bacterium, add the tailoring chemistry that makes the molecule a drug, and then start swapping parts to generate new candidates and read structure against activity. With antibiotic resistance rising and the discovery pipeline slow, having a fast-growing lab organism that can be reprogrammed to churn out variations of macrolide antibiotics is a genuinely useful place to start looking for the next ones.